Okay, good morning, good afternoon, good evening, everyone. Thanks for coming to the Spatial Omics Series 7 actual edition of last talk. I'm very, very excited about. We're delighted to have Dr. Amjad Askary from University of California, Los Angeles to give us the seminar today. And Amjad is currently assistant professor at UCLA in the Department of Molecular, Cellular, and Developmental Biology. And he's also a member of the Institute for Quantitative and Computational Biosciences, and also the Brain Research Institute over there. So Amjad is very interested in the study of how cell fate decisions are orchestrated during the developmental process, and has been developing numerous molecular tools that can help us better answer this question. So I think Amjad did his undergrad research, got his undergrad degree in Iran at the University of Tehran. And then he moved to US working towards his PhD degree in the laboratories of Dr. Gay Crump at USC. And afterwards, he decided to stay basically in the same area and moved to Caltech and working with microallowates, working on sort of the cell fate decision and the system biology. And after his postdoc, I really think he loves this kind of Angel City so much, and move across town back to the kind of ocean side of LA and join the faculty of at UCLA. All right, so without further ado, and we really look forward to the talk. And yeah, and the floor is yours.
Yeah, thanks for the introduction. And I also want to thank you and Ahmet and everybody else that is involved in putting together this seminar series. I'm personally really excited and optimistic about the future of spatial biology. And I think this the Spatial Omics seminar series has played a major role in spreading the word and creating a community. So very glad to be a small part of it today. As you guys know, the bulk of this seminar series has been on profiling or mapping gene expression. But for my part, I want to basically talk about something slightly different, talking about imaging-based approaches to read out barcodes, which is something that myself and a few friends and colleagues have been working on for the past few years. So, okay, yeah. To begin with, I want to remind everybody here that biology is dynamic. Cell division, lineage commitment, cell fate choices, pretty much everything that we study in biological systems happen over time. So it's very important to be able to capture this dynamic and record it to understand the rules that govern these processes. And the major tool that we've had for capturing the dynamics in biology so far has been time-lapse microscopy, which is a very powerful technique, and we learned a lot by looking at embryos and biological systems evolve over time. But it has some fundamental limitations. One of these limitations is that it's limited to systems that are optically accessible and you can image on a microscope. Also to the processes that happen over a time span where time-lapse microscopy is feasible. And also the number of channels, the number of different things that you can look at the same time is limited to the fluorescent channels that you have and can distinguish. Our ability to study dynamics in biology has been somewhat limited compared to other areas where over the past decade or so, we've had huge progress in terms of characterizing the system. So if you want to characterize a biological system at one time point, we have great tools like single-cell RNA sequencing or spatial transcriptomics that allow us to measure pretty much all the genes in the cell at one single time point. And it's exciting to think about how we can leverage this new ability toward studying dynamic as well as studying things over time or the history of the cells as well. And one of the ways that we can possibly approach this is through genetic barcodes. So barcodes have already been used in a lot of applications in biology and biotechnology. For example, if we label cells with genetically inheritable barcodes, we can do clonal analysis. We can look at what type of cells they make. Or if we label different perturbations by different barcodes, we can do multiplex screening, for example, CRISPR screen. And more recently, a new type of barcoding has attracted a lot of attention that I like to call dynamic barcoding or molecular recording. And in this case, barcodes evolve over time. They acquire changes over time. And this allows us to basically look at the relationship between cells or the history of the cells in more detail. The basic idea here is pretty simple, even though the techniques might seem a little bit fancy. The basic idea is that using genome engineering technology, we can basically look at the piece of DNA in the genome of the cell as one bit of information. It can be in one state initially, we can call it 0, and then using genome engineering tools, we can change it to another state, so from 0 to 1. And this can be done with any number of tools that we now have to edit the genome. For example, it can be done by CRISPR-Cas9, by integrases or base editors, but all of them basically do this basic operation. And if you have a number of these zeros or number of these barcodes in an array, and the changes in this array happen over time stochastically in this array, then you can use the state of these arrays at the end point of the experiment, basically at the final stage, and infer the relationship, the lineage relationship between the cells. So for example, in this case, we have one mutation happening in this barcode and another mutation happening in this region. And because these two cells share the first mutation, we can infer that these are more closely related to each other compared to these other two cells where they share a different mutation. And that's the basic idea behind lineage recording or lineage reconstruction. Similarly, if the edits that happen in the barcode array are proportional in their rate to some signal, then the clades or the lineages where you have more of that signal will acquire more edits, for example, in this green area. And the lineage where there is less signal acquire less edit. So again, by looking at the endpoint, looking at one time point, a snapshot, you can infer what happened to the cells in the past. And this basic idea has been very appealing to a lot of different researchers in different labs in the recent years. So now we have a variety of methods that leverage this basic idea to either record the lineage relationship between the cells or the signals that the cell receive. And here I'm just showing a few examples of these. I'm sure that by mistake or forgetfulness have not included all of them. But all of the methods that I'm including here using sequencing as the readout. And sequencing is great and it's very powerful. First of all, it's very accessible. Many labs can try it. Does not require spatial, know-how or technology. It's basically commercially available, and also it's very scalable. But it has some basic limitations. And one of them is that you need to dissociate cells for sequencing, so you lose the spatial aspect that is near and dear to all of us. And also the capture rate or the recovery rate for this barcode is not great. So what happens is that the lineage trees that you get from these approaches, one of them I'm showing here on the right, are not exactly similar to the lineage trees that you get by time-lapse microscopy in the sense that you don't have every cell division. You can think of them as an actual lineage tree where some random nodes in the middle have collapsed. So you can have like one cell giving rise to many cells, basically. For us, the goal that we basically try to achieve and we set for ourselves is to develop tools that allow us to look at the section of a tissue, and by looking at it, not only be able to tell the state of the cells at the final point where the tissue was fixed, basically the gene expression of the cells or the morphology or the position of them, but also be able to infer something about the history of these cells, how they're related in terms of lineage, and what signals or what level of signals they received in the past. This is an overall goal. By no means I claim that we already achieved this goal, but this is where we want to be, and we're kind of working toward it. And the first approach, or the first basically proof of principle for a system that allows us to get to this goal was published in 2017. It was called Memoir. And at the core of it, there were these barcoded scratch pads. So they have two components. One component is a scratch pad, which is the target for Cas9, repeated target, 10 repeats in this case. And next to it, there is a sequence which makes each individual integration of this scratch pad into the genome unique. So there's a barcode sequence next to it. And this whole sequence is transcribed in the cell as mRNA, and then can be detected by FISH or fluorescent in situ hybridization. And you can, using piggyback transposition, integrate multiple copies of this barcoded scratch pads together with other components that you need for editing them, including Cas9 or gRNA into the cells. The important feature of this barcoded scratch pad is that they have two states, of course. They have an intact state and a collapsed state, again, 0 and 1 that I mentioned earlier. And these two different states can be distinguished using FISH or fluorescent in situ hybridization in a way that the intact barcode will generate signal for both barcode and the scratch pad, and the collapsed barcode or the collapsed scratch pad will generate signal only for the barcode area, not for the scratch pad. So in this way, you can kind of distinguish which state the barcodes are using imaging and not sequencing. And then using sequential FISH, which you probably heard about in one of the previous sessions of this seminar series, you can read multiple barcodes and put all the information about which barcode is collapsed and which barcode is intact together to kind of infer the lineage relationship between the cells. So the group that was working on this and developing it, they noticed an issue, which was when, so the expression of these barcodes into mRNA depends on how many integration you have in the cell. And here I'm showing the number of DNA integration site and the relative RNA expression in these two graphs. And you see a big correlation between the two of them. What this means is basically when you have just one copy of a barcoded scratch pad integrated into the genome, you don't have high expression, and it's very hard to detect these barcodes. And this becomes a problem because you kind of need multiple copies of these in the genome to be able to detect it. But then when you have multiple copies, some of them might be intact, some of them might be collapsed. And this results in the readout not being digital and not being very straightforward to say which barcode is collapsed and which barcode is intact. So the group working on this and developing it looked into this very carefully and characterized it very well. And they showed that actually the main source of noise in the Memoir experiments is this variability in expression. But nevertheless, even though you have this uncertainty about the state of the barcodes, the system was still good enough to reconstruct some lineage trees, and it can be used to infer interesting things about cellular dynamics. For example, the dynamic of gene expression. In this example, they looked at the ESRB expression, which is bimodal in the stem cells. These are mouse embryonic stem cells. And by reconstructing lineage trees and looking at the state of the ESRB expression at the endpoint, they were able to infer the rate of transition between an ESRB negative state to an ESRB positive state. So altogether, this first version of Memoir was a great proof of principle that showed the potentials and also the challenges of having an imaging-based method for recording the history of the cells. The main drawbacks here were, as I said, was that the readout was not digital, it was noisy. And also, because they were using CRISPR-Cas9 for editing these barcodes, you could not put them in an array. If you want multiple barcodes, you needed to put them in different places in the genome, because if you have multiple double-strand breaks near each other, you will have a collapse in the array. And also, because they were using FISH for detecting edited and unedited state or collapse or intact state, they required large deletions in the scratch pad for it to be detectable by FISH. And this is basically the context of the problems that we were dealing with when I joined the team and I started working on this. And these were the issues that I wanted to solve. So let's for a minute go a little bit deeper into what the issue here is. The main issue of barcode readout when it comes to imaging-based methods is that, especially the hybridization-based methods, is that for us to be able to get a reliable signal from any mRNA in the cell, we need multiple probes to bind to it. Each one of them contributes a little bit to the fluorescent that we detect by microscopy because it has a fluorophore attached to it. And together, multiple of these probes together can produce enough signal above the background noise for it to be detectable. This is usually not a problem for mRNA detection because mRNAs are usually big enough that can accommodate these multiple probes. But for barcode readout specifically, this is a big problem because we want barcodes to be as short as possible. And when you need 20 to 40 probes, and you will need at least 500, maybe 1 kb size as your minimum detectable size, which is too much for a barcode. And also for expressing them, when these barcode sequences that are usually repetitive gets larger and larger, it's hard to make sure that they're expressed reliably in the cell, as I showed you in the first version of the Memoir paper. So these are basically the limitations that we're trying to solve. And if you think about it, you realize that all the edits on all the information in the barcode is in the DNA form. This transition from DNA to RNA, we only needed to be able to efficiently detect it. So it's kind of it's not fundamentally needed for encoding information into the genome. So the solution that I came up with for this problem was to use phage RNA polymerases. These are very robust enzymes that can detect a specific promoter sequence, a small promoter sequence, and transcribe DNA downstream of that promoter sequence. I'm sure most of you are already very familiar with them because they're used in a lot of different applications. These polymerases are not present in animal cells or mammalian cells that we work with. So if we engineer the promoter upstream of the barcodes, these barcodes are not expressed when the cells are alive, but they're in the genome, so they could be edited if we want to. And then after all the barcoding and editing is done, we fix the cells and add recombinant phage RNA polymerase to the cells, which will then find the promoter and transcribe the DNA. So the transcription happens after the experiment is done. And then under the right circumstances, you can keep a lot of these transcribed barcodes in the active site of transcription. And in this way, kind of amplify your signal. So this whole approach basically gives us 3 advantages over transcribing barcodes as mRNA when the cells are alive. And one is that it's independent of endogenous expression. So if doesn't have to be cell type specific and have the variability that endogenous promoters have. Second, as I show later, it enables detection of shorter barcodes, up to 20 base pair, and also enables discrimination between these barcodes based on single nucleotide variations. So I'm going to basically go through this and show each one of these items. So first of all, we wanted to see if this idea even works. So we made a mouse embryonic stem cell line that has one integration of a sequence that we can fish for downstream of these phage promoters. It has one and only one of these integrations. And when we add each of these three different phage RNA polymerases that we use in our studies, we can see that only in the condition where you have both the promoter and the polymerase, you get the transcription, which is what you expect. And these bright dots are basically the active site of transcription. So the basic premise works. You can transcribe them after fixation of the cells. But what we noticed was that even though, as I mentioned, integrations are verified to be one and only one per cell, not all the cells have the active site. So in the case of T7 and T3, about 85% of the cells have the active site, and SP6 is a little bit lower. We call this percentage, the 85% or whatever number that you get, the efficiency. Basically, it means the fraction of barcodes that are present in the system that we can detect. And the fact that it's not 100% is important for us. So we wanted to kind of look into it more carefully and find out why that is. You can think of basically two possibilities. It can be something at the cell level. For example, the cells are not permeabilized. Some of the cells are not permeabilized. Or it can be something at the barcode level. For example, some of the promoters might not be firing at the specific time that we fix the sample. And to distinguish between these two scenarios, what we did was making another cell line where we have three barcodes this time, each one having its own promoter. And again, the cells have one and only one integration of these barcodes. The idea here is that if it's something at the cell level, you expect a correlation between whatever you see for these three different barcodes, because all of them are in the same cell. But if it's something at the barcode level, there doesn't need to be any correlation between detection of these three barcodes. And what we saw, in fact, was we didn't see any correlation between them. So here I'm showing the probability of detecting barcode 1 and then condition on detection of barcode 2 and 3 and all the different combinations. You see that there doesn't seem to be any sort of correlation between them. And this is important for us because it means that whatever is limiting the detection is happening at the level of promoter. So to compensate for it, we can add more promoter and barcodes. And that's what I'm showing here, that by adding 3 of them, for example, we can increase this efficiency to 98%, basically, from 85 to 98%. So it's basically arbitrary, depending on application, depending on how much tolerance you have for misdetection, you can increase this number and get the level that you want. So far I just talked about detecting barcodes. Now, can we decrease the size as I promised? And the idea here is that instead of using the length of the transcript, as I showed earlier, for bringing together multiple probes, here we use the abundance of transcript in the active site to bring the probes together. So all of these red probes, for example, are designed against one 20 base pair target, but because there's multiple copies of these 20 base pair target in the active site, it brings together multiple probes and it amplifies the signal to the level that we need for detection. And the same thing here for green and yellow and so on. So this is basically what we tested here. The 3 probes, you can see that for each one of them that is 20 base pair long, you can see a clear signal. And then the signal for these 3 overlaps with each other, which is important because it tells us that we're actually looking at the barcode, not at some noise or some random dirt, for example. And that happens for all the different RNA polymerases that we tested. this is exciting because what it means is that you can now concatenate this much smaller sequences, 20 base pair sequences, together to make a combinatorial barcode library. So the idea here is that you have, for example, in this case, 12 sites or slots, basically, which each one of them can have one of 4 different possibilities. And then together, all 12 of them will have 4 to the power of 12 potential combinations. And the number of combinations here grows exponentially, whereas the number of sequences that you use and therefore need to detect grows linearly. So for example, for 16 million combinations, you need 48 fragments that you need to detect. This is very similar to much more familiar sequencing-based barcode libraries, where each one of these 12 is just one nucleotide and can have 4 different states, basically. But this one, you can image it instead of just sequencing it. And this is pretty much what we did. We generated barcode libraries starting from 81 and then 4,000 different combinations. And now the most diverse one is 1.5 million different combinations, which at this point, the diversity of barcode combinations basically is equivalent to what we have for sequencing. And here in the plots on the bottom, I'm showing the distribution of the different combinations in the library. So this sharp decrease in the beginning means that some of the barcodes in the library have higher frequency, but then a lot of them have, a more or less similar frequency, which is what we need for uniquely labeling the cells. And just similar to the previous version that I showed earlier, you can read this by sequential fish. You can read the state of each one of those, for example, 6 or 12 different states with imaging them by different fluorescent colors and then redoing it in the next round. We also confirmed that at least for some of these barcode libraries, we've done the confirmation that what we get from the imaging-based approach is comparable with what we get from sequencing the same barcode library. So now we have this barcode libraries that are image readable. We can use them to label cells, and also because these are pretty small, you can easily package them in the lentivirus. So we can deliver them to the cells by lentiviruses and label the cells during development and then read out these different combinations and find out which cells are related to each other or not. So here I want to basically do a quick detour and go to the biology side of it and say what we want to do with this barcode library. And hopefully kind of stimulate thought for you to also think about what you can do with them. So for me specifically, because I'm coming from a developmental biology background, I'm interested in development. And specifically right now, we're focused on development of the retina. So in the retina, all the different neurons and glia are made by retinal progenitor cells that are labeled here in green. These are multipotent progenitor cells, so each one of them can give rise to all the different types of retinal neurons and glia. And if you label one of them in an early stage of development, the clone that they make looks like a column. So in this case, a relatively larger clone is shown, just to show that there is not a lot of dispersion of cells, that clones look like column. But in terms of size of these clones and the type of cells that they're made of, they're very variable. So here I'm showing the distribution of the clone sizes. You can see that clones can be anywhere from just a couple of cells to a couple of hundred cells. There's a wide variety of different sizes, and also there is a wide variety of different combinations of cells that the clones can have. However, somehow during development, these variable clones come together and make a very stereotypical and regular structure in the retina, which is required for the function of the retina. So this is kind of a schematic to show what it means is that you have different clones, different sizes, different cell types, and they don't disperse it. There's not a lot of cell sorting but somehow together, the structure that they make has the right number of all the different cell types that the retina needs. And this is kind of a mystery of how this works, that neurodevelopmental folk have been working on for a while. There is, to kind of simplify, maybe oversimplify things, there are two models that people think about. One model is that the progenitors are equal with each other in terms of their developmental potential. So they're not different from each other. They just go through different competency states. And in some of these states, they make a differentiated cell type. For example, they make a horizontal cell type or a cone. And another model is that the progenitors are pre-patterned, or they have fate biases to begin with. And some of them are prone to make certain cell types, and some other ones are prone to make other cell types. And the distinction between these two models is not purely academic, although it is somewhat academic. It's also important because when it comes to making these different cell types for transplantation, for example, you want to know how they're made. If they're made by pre-specified progenitors, you want to know markers for those progenitors to be able to isolate them and make the cell type that you need. So in a lot of neurodegenerative diseases, for example, in glaucoma, retinal ganglion cells die. So if you want to replace them by transplanting new retinal ganglion cells, if there are progenitors that are pre-patterned to make this cell type, you want to know that. Whereas if you have equal potential and basically random assignments of cell type, that affects your strategy for making different cell types, basically. So here I'm basically just showing a preliminary analysis of clonal composition in the retina. So we're clustering clones based on the number and type of cells that they have. And what we saw is that there's at least 7 different groups of clones or clone types in the retina. And some of them include those clinically interesting cell types. So for example, retinal ganglion cells are only found in one of these different clusters. So this by itself is basically kind of suggesting or supporting the idea that different progenitors might be pre-specified, although it's not, a very direct observation. And also when we look at what drives this clustering here, we find some correlations that make sense based on the literature or are previously known. For example, we find a correlation between cone and horizontal cells. And we know that there are specific progenitor cells that are biased toward making these two cell types. We also find correlation between rod and bipolar cells. And that makes sense because both of these cell types are born later in the development of the retina, and they're very frequent. So it makes sense to find more of them together in the same clone. But we also find certain correlations, for example, between molar glia and amacrine cells that, at least to our knowledge, is not expected or is not explained by anything in the literature. So we find some new correlations that we can then go and investigate further. So basically following up on this, we want to ask more specifically whether the retinal progenitor cell types are, whether there are specific progenitor cell types that, for example, make ganglion cells or not. And also look at the distribution of these different clone types in the retina, because when we're doing clonal tracing with the image readable barcodes, we also know where these are, so we can ask how they're distributed across the retina. And also further investigate these correlations that we find. So this is kind of the type of things that can be done if we have a scalable lineage tracing technology. So far, I basically talked about one-time labeling, meaning that at what time point you introduce barcodes to the cells, and these barcodes don't change anymore. But what I showed initially, or what I introduced as our goal, is more than that. We're also interested in finding the relationship between the cells and kind of the more detailed structure of the lineage trees. And I mentioned earlier that this can be done by editing barcodes over time, and there are different ways to do it. I have a favorite between these different methods, and that is base editors. The reason for it is that CRISPR, as I mentioned, creates a double-strand break that is problematic for barcodes. If you have an array of barcodes and you have multiple double-strand breaks, it might collapse the barcode. And also integrators have size limitations, so you can't reduce the size beyond a certain level. Whereas base editors can introduce precise edits, even up to one nucleotide changes in the genome, and the changes are predictable. So in case you're not super familiar with them, these are basically a new tool in the CRISPR toolbox that are made by fusing a mutated Cas9 to a deaminase. And the Cas9 guide RNA basically bring the deaminase to a specific site in the genome, and then open up the double-strand helix, and then deaminase changes one of the nucleotides. And there are different versions of it. It can change from A to G, which is the one that we use most often. It can be other changes in the genome. So there isn't any double-strand break involved. The change is a small change, and it's predictable. It's pretty much, in my opinion, the perfect tool for writing information into the genome. The problem is for imaging-based approaches that these small changes are not easy to detect, or at least haven't been easy to detect previously. So one of the things that we wanted to explore was that, can we use this increase in sensitivity that we get from accumulation of transcripts in the active site to distinguish between these single base edits? And kind of to show the idea here, we can have a mix of probes that are identical to each other except for one base pair. And if you hybridize a sample with these probes, the one that is a perfect match for the target is going to bind to most of the transcripts in the active site, whereas the rest of them might be mostly washed away. So there will be a difference in the signal intensity between the one that is the right match and the ones that are not, they're mismatch. And this is basically what is shown here. So in this case, the target has an A, so the right probe has a T, and each column here is one cell. You're looking at different probes that have T, C, G, or A in them, and different colors. And you see that in each case, the probe that has the right nucleotide produces the majority of the signal. So in each case, T basically gives you the majority of the signal. And it's very easy to see in this one example, but we also quantify it over many examples. And as you can see here, you can very reliably tell that the target is an A because the T probe binds to most of it. And we also looked at other bases and other positions within the barcode, and pretty much all cases except for the first position, you can very reliably detect a single nucleotide change within a 20 base pair barcode. So these are all in cell culture. We also want to test it in tissue. So what we did was to, again, package this in lentivirus and then inject them either in the mouse brain or in the chick embryos. And in both cases, qualitatively, you can see the same thing, that the match probe produces a majority of the signal, regardless of which channel it's in, and regardless of what type of animal or embryo we're looking at. And also to kind of quantify it, we use two viruses. Each one of them has two barcodes. So barcode 1 here are the same sequence between the two viruses, except for this one position. That is A here and G here. And barcode 2, again, the sequence is the same except for one position. So the way this quantification here works is that if we're detecting it correctly, for each inactive site, we need to detect either both A or both G, because that's how the viruses are made. And if you see GA or AG, that's a misdetection, basically. And as you can see here, in the majority of cases, in fact, in 92% of the cases, we correctly detect the combinations. So both in cell culture with higher accuracy in cell culture, but also in the tissue, we can detect single nucleotide changes within the 20 base pair barcode sequences. And because that was promising enough, we went ahead and generated barcode arrays where in each element or each unit of the barcode array, there is a target for guide RNA, and there's one specific nucleotide A that upon acting of the base editor can be converted from A to G. And then next to each one of these gRNAs, there is an ID sequence that distinguishes between these different barcodes. So there are 12, for example, in this case, 12 different ID sequences. Again, you can think of this as the array of 0 and 1 that I showed initially. So all of these are initially in one state and then can be switched to another state through the action of the base editor. So I made two versions of these. One where each one of these barcodes is targeted with a different gRNA. This can be useful if you want to record multiple channels or you want to have, for example, multiple signals. And another one where one gRNA targets all the barcodes in an array. So this is more useful if you want to have more diversity generated by a single gRNA. And in both cases, you can see that between the edited and the unedited cells, visually, it's very easy to distinguish them, but also quantitatively, we can cluster them relatively easily. And as you expect, you only see the edited when you have all the components present, the base editor, the guide RNA. And this is shown here for one of the barcodes and shown here for all of the barcodes. And also quantified in terms of how confident we are for detection of the edited versus unedited, and how correlated it is between doing it by imaging or doing it by sequencing. So basically at the moment, one focus is to generate stable transgenic lines, mouse lines, that have these barcode arrays in them, integrated in different places in the genome, so that we have more targets and more recording capacity or memory. Each one of these arrays has an ID barcode that can distinguish between the ones that are integrated in different genomic loci. So this will be basically a very powerful tool for studying lineage using imaging-based readout, but the stable transgenic lines are also have their own downsides because they take a long time. And alternatively, we also generated libraries that are good for transient transfection or electroporation. So in this case, they work with piggyback, and you can electroporate or transfect both the base editor and guide RNA and the barcodes into the cells. And we showed that you get multiple integrations per cell. And this way you can basically do experiments kind of quick and dirty. And all of these tools, and as they come online, we're very happy to share with anyone who's interested in using them. And one more small thing is that edits that happen in this recording arrays can be used for basically inferring the lineage relationship between the cells, as I showed in the initial Memoir paper, but also can be used for inferring signaling. And this is actually an area that I'm really interested in, because the level of the signal is basically encoded in the number of edits that you have in the barcode arrays. It's also shown here by simulation. But also specifically because we're using base editors, it gives us some additional capabilities, some additional kind of clever things that you can do because the edits are predictable and single or two nucleotides or one nucleotide, depending on how you arrange the base pairs. So one of the things that, for example, you can do, you can make composite barcodes. I'm showing an example here. I'm going to kind of looks a little bit complicated, but it's actually not. And the way it works is that, let's say you want to detect the order of two signals, X and Y. And signal X produces gRNA, the red gRNA, and signal Y produces the blue gRNA. So you can make your barcode so that the PAM site, the NGG site, for the first signal is intact the way it should be. And the PAM site for the second one, for the Y, is broken. So it's NAG or NGA or NAA, some broken PAM site. But it's positioned in a way that the first signal will fix the second PAM site. So what happens here is that if you have the first signal, it fixes the PAM site for the second. And then later on, if you have the second signal, it can make the edit. And this second edit is what we read out. So essentially what happens here is that you have a composite barcode that detects X and then Y. If you have signals in this order, one signal and then the second signal, it will be edited. And if you have the opposite order, it will not work. And this is basically kind of a preliminary result showing that, in fact, the second edit only happens when the defective PAM site is fixed. So just basically want to emphasize the fact that using base editors gives you the possibility of doing more tricks because the outcomes are predictable. Okay, so to summarize everything, I talked about essentially three different types of things that we can do using barcodes. All of these either are possible using sequencing or have been possible for a while using sequencing. But specifically, we're trying to make them imaging compatible so that we can capture the spatial context of the cells, the morphology of the cells, and other information that are relevant to a lot of biological systems, especially the nervous system. The first one was lineage tracing using combinatorial barcodes that can be imaged by microscopy. And the second one was edits in the barcodes that can encode information about the lineage relationship between the cells. And going forward, as I said, we want to have a stable transgenic line. We also have tools for doing this experiment in transient. And then kind of in future, or our future direction is to work more on the signal recording. And both the intensity of the signal that can be encoded in the number of edits and also the order of the signals that can be encoded using base editors. And with that, I want to thank everybody involved in the work that I showed today, both my previous lab, Elowitz lab, Michael, and the students and postdocs and technicians that I worked with there, collaborators in Kai Lab and Lewis Lab. And also my new and growing lab here at UCLA. And obviously we're hiring at all levels. So if you're interested in kind of working on any of these things or have ideas along these lines, feel free to reach out. And thanks again for your attention.
Yeah. Thank you, MJ. This is super exciting. So also complements very well most of the seminars in the spatial omics seminar series. I think we have been talking about sort of the dynamics in the spatial biology, how to record it. I think you'll really provide I think the most powerful tools people can imagine right now and down the road. I'm sure they're gonna approach you and adopt your technologies in the spatial biology space. So I have like already quite a few questions in the chat box, but I just want to ask my question first. So talk about like a recording. So can you also kind of perturb the cells, kind of combine this recording, but since I think the last part of your talk, there's like a two-step so recording, so you can have one signal to trigger the other. Maybe the other signal you're going to trigger can kind of induce something, for example, T cell activation.
Yeah, that is that is actually something that we've been thinking about, and we're very excited about. So the idea is that like recording, in the sense that I talked about, is writing something in the genome that we read later on and interpret. But we can also think about writing something that the cells read and interpret for themselves. I think for that application, maybe integrases are a better choice, because it's easy to kind of, for example, through inverting or deleting, perturb the function of a gene or overexpress a gene. But specifically for base editor, also we've done a little bit of work. The basis of it is that you can activate or inactivate guide RNAs through edits that are happening from other base editors. So it's basically a cascade of guide RNAs. So you have one guide RNA either activating another inactive guide RNA or inactivating an active guide RNA. And because guide RNAs can be used to modulate a lot of different functions in the cells, this can be potentially pretty powerful. Guide RNAs can be used to, activate, inactivate other genes. So this is something that we're exploring. I didn't show anything about it here today, but I don't know, hopefully in future.
Yeah, I think the future is really unlimited.
Yeah.
Yeah, I think quite a few questions in the chat box now. And the first one from William Allen. He was asking to increase the efficiency of the zombie readout. So why concatenate multiple barcodes and then not multiple promoters?
Yeah, so multiple promoters did not work. We tried it. I think, so we're not really sure. So I showed some data that shows it's happening at the barcode level or at the promoter level, not at the cell level. But we're not really sure is why it is or what is exactly happening. One thing might be that there are damages to DNA, for example. Some portion of the DNA might be broken during the fixation phase. And if that's the case, then adding more promoters is not going to help, because if part of the barcode is broken, more promoters is not going to fix it. Another possibility is that just firing of the promoter might be a stochastic process with a certain frequency. So there's nothing wrong in this scenario. There's nothing wrong with anything. It's just that each promoter happens with a certain frequency and you're fixing it at a single time point. So you're likely to capture a fraction of the promoters. And if that's the case, the only way to kind of overcome it is to have more of promoter and the barcodes. Or, I don't know, change something that increases that frequency of the firing. So basically the short answer is just adding more promoters is something that we tried and did not help. If you're, and also so far, we've really, at least in maybe others have noticed the need, but for myself, I have not noticed the need to kind of increase the efficiency much higher than what we're already getting.
Okay, so next question from Lin Zhang, and a great talk. Okay, could you elaborate more on a retinal clone and correlation of cell types in a PCA plot and the perspective of applying this barcode technique to retinal progenitor lineage tracing?
Yeah, let me go back to be more clear. Okay, so what is shown in this plot, each point is a clone, and each clone has a certain number of different cell types in it. So usually when you're looking at these UMA plots or PCA plots, you're looking at each point being a cell, and each cell has different genes. Here, each point is a clone, and each clone has different cells. So that's why I called it clone type, not cell type. So these are clusters based on the number and the type of cells that they have kind of mathematically, it's exactly the same process that you do for single cell RNA sequencing, but the numbers are coming from cells, not from genes. So hopefully that makes it a little bit more clear. Yeah, and then in terms of prospect of like going forward, what we want to do with this, one of the interesting things is that because we know where these clones are because we have the images basically. We can look at the correlations in the space. So because again, going back to this problem of how do you regulate the composition of different clones to get the right number of cells at the end, traditionally, these type of things have been done through lateral inhibition. So one clone signals to the clones around it that if one clone makes too many of a certain cell type, signals to clones around it to not make as many cell types. If this is the case, you expect to see anti-correlations or negative correlations between clones that are closer to each other. So these are kind of the type of analysis that you could do using this method that are not possible by other methods of clonal tracing, because, for example, if you're doing GFP labeling, clones that are too close to each other, you can't really distinguish between them. So, so this is kind of the type of things that you can do with this.
Yeah, thanks. So next question from Peng Fei. The question is, okay, so first of all, nice talk. And could you further elaborate more on a comparison between CRISPR-9 system and a base editor? Is there any limitation of using the base editor for embryonic development study?
Yeah, okay, so molecularly, the difference is that in the base editor case, the double strand is intact and the change is being done through deamination. Whereas in the CRISPR case, you make a double strand break and then the changes, insertions and deletions happen through the non-homologous end joining. As far as the implications for the technical side of it, is that if you have double strand break, if you have two double strand breaks near each other, whatever is in between the two might be completely missed. And that happens a lot in, for example, approaches that use CRISPR for barcode, like for example, in Gestalt. If you look at that paper, they quantify how many deletions they have that delete the barcodes in between. And deleting barcodes is not good because that removes the information. As far as development is concerned, there are concerns that overexpression of these deaminases in the cells might be toxic because these deaminases might have off-target effects. It might work on the RNA of the cells. So double strand break is also not something that cells specifically like. But so far it seems like the toxicity might be something if you want to overexpress them for a long period of time might be concerned about. At the moment, I don't think there is any stable transgenic mouse line that overexpresses base editor. So we don't have a clear answer to that. But also, at least in our hand, for overexpression specifically in the retina and the level and time frame that we're working with, we also have not seen overt cell death. So an area that we're kind of aware of, but need to be studied later when we have a transgenic mouse line that expresses base editor.
Yeah, so great. I think next question about the T7 RNA polymerase. So after you fix the cells and introduce the T7 polymerase, are all the transcribed RNAs gonna stay where they were transcribed, or have you seen the transcribed RNAs kind of float around?
Yeah, they do float around. I mean, they're kind of like schematically showing them here floating around. But yeah, they do float around. But if you're using fish, not HCR, so basically, so if you're doing HCR, hybridization chain reaction, I hope you might be familiar with, that's what you see. So those RNA molecules that are floating around are detectable using HCR. If you're doing fish, which I think is this case, you can't see them because there is no signal amplification. In this case specifically, we're using a single probe. So the signal generated by those individual molecules that float around is not high enough for you to detect it. So essentially, they don't create any problem in terms of barcode readout for us. We would, of course, I would love to keep as many of them in the active site as possible. But the reality is that they do float around, and also they go to other cells. That's also something that we showed. So you can't really use them for labeling the cells. You can only rely on the active site. In case of, even if you do HCR, the intensity, it's kind of in the supplement of the paper, that the intensity of the active site and the individual RNA molecules shows a bimodal distribution. So you can always tell which one is the active site, which one is the floating around RNA.
So in that case, can you kind of fix the cells one more time, and then you can kind of see better where those?
Yeah. So I should say we tried a few different things, but not exhaustive. So there might be still other things that we haven't tried, especially for the case of tissue sections. I think there might be things that can help, like using a hydrogel matrix that has, for example, LNA probes that capture RNA, things like that. Under construction. We're working on it.
Yeah, also right now that doesn't cause any problem, right?
Yeah.
Yeah. So I think in the interest of time, just a last question in the chat box from William Allen again. So does T3 produce more RNA, or is the intensity scale different between T7, T3, SP6?
No, the intensity scale is the same. T3 does produce more RNA, and that is kind of the enzyme that we currently using, even though initially we tested multiple of them. Right now we're using mostly T3. I'm not sure if it's inherent to the polymerase though. It might be that just the enzyme is more concentrated because we buy these as recombinant proteins. So, so it might be something specific about T3, or it might be something just in the prep, but it does produce more RNA.
Yeah. All right. So thank you, Amjad, and also thank you, everyone. This amazing talk and also amazing topic. So I think you are developing technology to record the history, but I really think this talk really leads to the future of the space, the spatial biology. So thank you very much, and thank you, everyone, and you'll have a good weekend.
Thanks, you too.
Thanks.